Welcome back to Fusion Facts. The Human Genome Project, Decoding the Book of Life. The Human Genome Project, HGP, stands as one of the most ambitious scientific endeavors in human history. Launched in the late 20th century, this international research effort aimed to unravel the complete sequence of the human genome, a sequence of DNA containing all the genes that make up our genetic blueprint. The successful completion of the HGP in 2003 marked a monumental milestone in genetics and medicine, ushering in a new era of scientific discovery and personalized medicine. Today we will delve into the history, significance, and impacts of the Human Genome Project, exploring its scientific objectives, technological advancements, ethical considerations, and the transformative role it has played in understanding human biology and disease. The Human Genome Project, a historical perspective. The idea of mapping and sequencing the entire human genome emerged in the late 20th century, driven by the convergence of technological advancements, scientific curiosity, and the recognition of its potential for medical breakthroughs. The HGP officially began in 1990 as an international collaboration between multiple countries, including the United States, the United Kingdom, Japan, France, and Germany, among others. Its primary goals were to determine the sequence of all 3 billion base pairs in the human genome, identify and map all the genes, and make this wealth of genetic information freely available to the scientific community and the public. The enormity of the task at hand was evident from the start. The human genome is comprised of approximately 25,000 genes, which are scattered across 23 pairs of chromosomes. Each gene carries the instructions for producing specific proteins, which govern virtually every aspect of human biology and development. Prior to the HGP, scientists had only sequenced a small fraction of the human genome, leaving the vast majority of our genetic code a mystery. Technological advancements, a race to the finish line. One of the key drivers behind the success of the HGP was the rapid advancement of DNA sequencing technologies. At the project's inception, the most commonly used method for sequencing DNA was known as Sanger sequencing, a labor-intensive and time-consuming process. However, as the HGP progressed, new high-throughput sequencing technologies emerged, dramatically accelerating the pace of sequencing and reducing its cost. The development of automated DNA sequencers, coupled with the miniaturization of reaction volumes and the parallelization of sequencing reactions, made it possible to sequence DNA at an unprecedented rate. The introduction of shotgun sequencing techniques, which involved randomly breaking the genome into small fragments and sequencing them separately, allowed for rapid assembly of the entire genome. By 2003, when the HGP reached its completion, the human genome had been sequenced with a high degree of accuracy. This monumental achievement marked the beginning of a new era in genetics, as scientists now had access to a comprehensive map of the human genome, a map that held the potential to unlock countless mysteries of human biology and disease. The scientific significance of the HGP. The Human Genome Project has had far-reaching scientific implications and has paved the way for numerous breakthroughs in genetics, medicine, and beyond. Understanding human variation. The HGP provided insight into the genetic diversity among individuals and populations. This knowledge has been invaluable in understanding the genetic basis of diseases, as well as human evolution and migration patterns. Disease gene discovery. The HGP accelerated the discovery of genes associated with various diseases, including cancer, diabetes, and rare genetic disorders. Identifying these genes has opened the door to more targeted diagnostics and treatments. Personalized medicine. The availability of genomic data has enabled the development of personalized medicine approaches, tailoring treatments and therapies to an individual's genetic profile for greater effectiveness and reduced side effects. Pharmacogenomics, Pharmacogenomic studies, made possible by the HGP, investigate how an individual's genetic makeup influences their response to medications. This knowledge has implications for drug development and prescribing practices. Functional genomics. The HGP laid the foundation for functional genomics, which seeks to understand the roles of specific genes and their interactions in biological processes, furthering our understanding of human biology. Comparative genomics. Comparing the human genome with those of other species has shed light on evolutionary relationships and the unique genetic attributes that make us human. Ethical considerations and challenges. While the Human Genome Project has been a source of immense scientific progress, it has also raised ethical concerns and challenges. Some of the key ethical considerations include privacy and genetic information. The availability of individual genetic information raises questions about privacy and the potential for misuse of genetic data. Safeguarding genetic privacy and ensuring responsible data sharing are ongoing challenges. Genetic discrimination. Concerns about genetic discrimination in employment, insurance, and other areas have prompted the need for legal protections, such as the Genetic Information Non-Discrimination Act, GINA, in the United States. Informed consent. Ethical guidelines for obtaining informed consent from individuals participating in genetic research are crucial to ensure autonomy and transparency in genetic studies. Equity and access. Ensuring equitable access to the benefits of genomic research and personalized medicine is a critical ethical consideration, as disparities in healthcare and genetic services persist. Responsible research practices. The rapid pace of genomic research requires vigilant oversight to ensure responsible conduct, including the handling of sensitive data and potential conflicts of interest. Ethical use of genetic editing. Emerging genetic editing technologies, such as CRISPR-Cas9, raise ethical questions about the potential for editing the human germline and the long-term consequences of genetic modifications. The legacy and future of the Human Genome Project. The completion of the Human Genome Project marked a historic milestone in scientific achievement, but it was by no means the end of genomics research. Instead, it served as a catalyst for further advancements and inquiries. Some of the ongoing and future endeavors in genomics include genomic medicine. Genomic medicine continues to evolve, with ongoing efforts to identify genetic markers for disease risk, develop targeted therapies, and refine personalized treatment approaches. Precision oncology. The field of precision oncology uses genomic information to tailor cancer treatments, offering the promise of more effective and less toxic therapies for patients. Functional genomics. Advances in functional genomics are helping researchers better understand the roles of genes and their interactions in health and disease, leading to new insights and therapeutic targets. Epigenomics. Epigenomics explores the modifications to DNA and associated proteins that can regulate gene expression. Understanding epigenetic mechanisms is critical for deciphering complex diseases. Population genomics. Population genomics seeks to understand genetic variation within and among populations, shedding light on evolutionary history, migration patterns, and disease susceptibility. Ethical and legal frameworks. Ongoing efforts are needed to update and adapt ethical and legal frameworks to address emerging challenges in genomics research, such as gene editing and data sharing. The Human Genome Project stands as a testament to human curiosity, collaboration, and the power of scientific discovery. Its completion has reshaped our understanding of human biology, disease, and evolution, opening the door to a new era of personalized medicine and genetic research. However, as genomics continues to advance, it also raises complex ethical questions and demands responsible practices to ensure the equitable and ethical use of genetic information. As we navigate the ever-expanding landscape of genomics, one thing remains clear. The journey to decode the Book of Life is far from over, and it holds the promise of transforming healthcare, our understanding of ourselves, and the future of medicine. Thank you for watching. Until next time.